In the second part of today's lecture, we again look on cooperative transitions, but now in a more chemistry view, and we will learn about cooperative binding and allostery. So going first back to the first part where we looked on protein binding, we had a physical interpretation of cooperativity, which is displayed as the steepness of the curves when we have a switch between two states. Now we want to look into a chemical analogon of cooperative binding, and that concerns the binding of oxygen to hemoglobin. Now, hemoglobin is the oxygen transporter in our blood. The oxygen is bound to iron, which are the yellow balls in this graph here, and hemoglobin comes as a tetramer, or 4 identical protein parts. Now, experiments have been done where you monitor the fraction of oxygenated hemoglobin, so how much oxygen has bound to hemoglobin, as a function of the outer concentration of oxygen that you allow to bind. The open circles are the experimental results. So it's again an S-shaped curve where you have very low oxygen binding at low concentrations and high oxygen binding at high concentrations. Just as a side mark, we analyzed the oxygen binding concentration just by a calorimetric measurement, so some spectroscopy, because hemoglobin would change its color on binding of oxygen. Now there is a similar protein in our body, which is myoglobin, which takes care of oxygen transport within or very close in muscular tissue. And that has a very different oxygen binding curve. There you see its steep rise to a maximum. So the aim now is to explain the observed behavior. So let's assume a chemical reaction. So we have a hemoglobin and there's oxygen, and oxygen can bind to hemoglobin. So it's a chemical reaction. Now the fraction of bound oxygen to hemoglobin is equal to the concentration of the oxygenated hemoglobin divided by the concentration of hemoglobin plus the concentration of oxygenated hemoglobin. Now that equation we just can rearrange, and we find that this is equal to 1 divided by 1 plus 1 divided by the equilibrium constant times the oxygen concentration. Now reformulating that, this is equal to the oxygen concentration divided by the oxygen concentration plus 1 divided by K equilibrium. Now for low oxygen concentration, we expect that this equation scales proportional to the oxygen concentration. But clearly, for the hemoglobin, this does not work. Yet it nicely describes the data for myoglobin. And recall, myoglobin was a monomer, whereas the hemoglobin was a tetramer. Hence, we need to repair our assumption. The main point is that we want to increase the steepness of the curve for the hemoglobin oxygen binding at the mid concentration. Well, we actually know how to do this. We need to introduce cooperativity. Now, the cooperativity that we now introduce is that we assume in the chemical reaction that, and knowing that hemoglobin is a tetramer, we now assume that the binding of oxygen to hemoglobin we rewrite as hemoglobin plus N times the oxygen gives hemoglobin bound by N oxygen molecules. So now we do the same analysis as we did before for N equals to 1. And we come up that the fraction of oxygen-bound hemoglobin now scales like the oxygen concentration to the power N divided by oxygen concentration to the power N plus 1 divided by K equilibrium. That scales at low oxygen concentration as oxygen to the power N, which gives this initial flat curve and then a steep increase. Now the parameter N that we introduced here is called the Hill coefficient, and it represents the cooperativity in the binding. Now for the particular experiments, N is not actually 4, but N is 3.1. So as a summary, cooperativity in chemistry is reflected or expressed as the Hill coefficient. And the Hill coefficient also in chemistry increases the steepness that actually is required in the case of the hemoglobin to lead to the required oxygen exchange in the lungs versus tissue. So in the lungs you need to acquire oxygen, whereas in tissue you need to release the oxygen again. When it comes to the reason why there is cooperativity in oxygen binding in hemoglobin, it actually comes to a surprise. If we look on the structure of hemoglobin with the tetramer, we see that the binding sites of the oxygens are in the order of 2.5 nanometer away. This is very large if we think about, for example, electronic interactions, where the wave function will die out within a few angstrom. So electronic interactions we can exclude, being the case for cooperativity. Now, a lot of studies have been done on hemoglobin, and what has been shown is that binding of oxygen to one of the sites will lead to major structural rearrangements of the protein. So helices will deform and shift along, and that results in a higher binding affinity of the open binding sites for oxygen on the other sites. So this effect is called allostery, where changes on one part of the protein will lead to structural and also functional changes at the very distant part of the protein. So let's look back in what we learned today in lecture 4. So we looked for protein folding, in particular the helix to coiled transition, and we realized that it is easily explainable by a two-state system, where we assumed some interaction of neighboring amino acids going on. The model that we used was the Ising model that explained the melting temperature of the proteins, but also introduced the cooperativity parameter that leads to a helical fold. Then in the second part we looked into chemical reactions. Also in chemical reactions we can express cooperativity, and we looked in particular for the cooperativity in oxygen binding on hemoglobin. And there we had to introduce a parameter which is called the Hill coefficient that describes interaction. And that comes as a power in the definition of the equilibrium constants. Further, we looked into the structural reason for the Hill coefficient in hemoglobin and defined the effect of allostery, where distant changes in the protein structure can lead to a change in the function of the protein.